Hello, Rajiv. That's a pleasure for me. Now he's not listening, like always. Rajiv, it's a pleasure for me that you have invited me for this excellent conference. I enjoy really being here. Yeah? If you simply look at the weather in Germany, it's cold and horrible, so that's fantastic to enjoy sunshine here and collect energy in Hyderabad. So I will talk about the emperor of the traits, grain yields, the most important trait, I would think, in breeding. And if you look at the past, you can observe that there was a tremendous increase in grain yield in different important crops, such as, for instance, wheat. And you observe a 1% increase in yield. And I would think that the key driver for this selection gain is sitting here. Yeah? So that was classical breeding, that was simply generating variation and then followed up by intensive phenotyping by identifying then better yielding varieties. And so that's very important to realize that this is such a key in improving yield. But now, if you look for the future, this yield increase is not enough, as shown nicely in that study. So we have to increase the selection gain per year tremendously. And that is difficult, so that means at least that we have to maintain the activities done by classical breeding. That's very important. But we have to search also for opportunities and innovations which allows us to boost selection gain. And very often it has been proposed that this is the key driver of the future selection gain. So exploiting genomics in breeding is hoped at least to speed up the breeding progress and ending up in a higher yearly selection gain. And the idea is often that you understand, that you deepen your understanding on the genetic architecture of the traits, and that you can then exploit this knowledge on the genetic architecture in an efficient way to do a kind of knowledge-based breeding. And I want to show you today some results which we performed in wheat. So we convinced one of the leading breeding companies in Europe, the KWS Locho company, to provide us with a large and comprehensive data set comprising 4,000 elite lines. And we could use these elite lines or this population for our studies. And you see here the structure, the genetic structure of these 4,000 elite lines, which have been fingerprinted with 15K SNP array. And you see a picture which is very typical for wheat breeding. That's a little bit unlike in maize, where you would have a very strong family structure of large biparental families. And this is not the case in wheat. You have a little bit of population structure similar like a diversity panel. And these 4,000 elite lines have not only been genotyped very intensively, but this panel of lines have been phenotyped also very intensively by the breeding companies. And you see here on the right side the distribution of the phenotyping intensity of these 4,000 lines. So some of the lines were tested in up to 17 locations. So the quality of the phenotypic data was on average yielded a heritability of 0.73, which is quite substantial. And if you look at the distribution for grain yield of these 4,000 lines, you will realize, if you look at the grain yield potential, that this is really a very high yielding germplasm we are dealing with, so that is really the best available. Okay, we took this genomic and phenotypic data and performed the genome-wide association mapping by applying linear mixed models, where we looked for significant additive effects contributing to grain yield. And we extended the models to do a two-dimensional scan also for epistasis. So we looked for genes talking to each other. And we found quite a substantial number of main effect QTLs, and this is illustrated here in these outer circles. You can see several points outperforming this Bonferroni corrected significance thresholds. And what is shown here in the inner circle is that we detected also in wheat that the genes are talking very intensively to each other, so that we have a substantial proportion of epistasis active in wheat breeding. So that proposes that it is very promising to perform this kind of knowledge-based breeding, that we could use and exploit this information to breed better varieties. But as we are Germans and we doubt that the results are perfect, we tried to enter a validation phase. And what we did, we took this data, which comprised actually line data from 3 cycles of selections. And what we did, we took the first 2 selection cycles, so 2,000 lines, which have been tested in the year 12 and 13, and we performed association mapping in this sub-panel of 2,000 lines. We identified several candidate genes under a very relaxed significance thresholds, and they are depicted here. Then we took this marker trait, or the markers, which showed marker trait associations in the year 2012 and 13, and looked for the p-values if we took the other 2,000 lines from the year 2014. And you can imagine that this result is quite frustrating. So the values are approaching 1, so there is no significant association left from all QTLs we looked for. So this is a very frustrating picture, which shows us that we don't understand too much on the genetic basis of this important trait grain yield, even in this very large population which we had at hand. So as we had data from 2 years, or 2 cycles, it is tempting to speculate that maybe genotype times year interactions played an important role, and leading to these non-significant associations if you look for the results across the years. And therefore, I've shown you here a set of overlapping varieties which have been tested in 12-13, but also in the year 14. These were in total 154 varieties. And you see that the grain yield performance of these 155 varieties were in good accordance across the years. So we observed a correlation of 0.8. So that means that genotype-by-year interactions played only a marginal role in this QTL detection. And that is really pointing to the challenges to identify key genes contributing to grain yield in wheat. So, and if you look for we looked for solutions, and there were an interesting proposal from the animal breeders that they said, okay, for such a complex trait grain yield, you need different models. It is unlikely that you have to identify key genes, but maybe all genomic regions contribute somehow to the contributions of grain yield. And studies from human genetics, for instance, have shown that you could do a much better job in explaining human height by using these models, so these genome-wide prediction models, which consider also genes with small effects. And it's shown here, if you shift from the methodology, from this genome-wide association mapping to this genome-wide prediction approaches, you decrease the missing heritability gap quite tremendously. But it seems to be really a solution also for breeding. So there was an interesting study published by Moivissen in the year 2009, and he asked himself the question, for these genome-wide prediction approaches, on how large should the population size be to do really a good job in predictions. And he wanted to explain 80% of the variation, and he investigated that based on theory and simulation studies for human populations. And he looked for different scenarios. So he investigated different heritability scenarios, ranging from very complex traits to rather simple traits in human beings. And he asked himself the question on how many markers do I need and how many individuals do I need in my population to do a good job in prediction. And he found, or he estimated, that he needs around 7 million markers to explain 80% of the variation in the global human population. That is quite high. And then he observed also that if the trait is complex, ending up in a low heritability, that you need around 11 millions of individuals to explain 80% of the variation. And if the heritability increases, you need lower population sizes, but you still end up with around 2 million individuals. And that would be even too large for the ICRISAT breeding program, I would assume. And that seems to be a little bit frustrating. So we took the parameters which Moivissen used and projected that into the wheat scenario. And the difference is that the effective population size in this global human population is much larger than for wheat. And there the picture looks then much more positive. So depending on the complexity of the trait, we need around 50,000 of individuals to explain 80% of the variation. And if you have a less complex trait, we can deal with around 10,000 individuals. And we need around 30,000 markers. So this is something which is in the range. It's still quite ambitious, but this is feasible. And then there's a further very important difference between human and plants. You can do much nicer things with plants. So, for instance, you can influence the heritability, which is not possible with humans so easily. So in plant breeding, we can clone the plant and not look at an individual plant, but we look rather at a plot. So we have 2,000 plants per plot in wheat. We replicate that. We replicate it not only in one location, but we use different locations. And there, with this approach, we can increase the heritability also for complex traits. And if we look at for cranial, for instance, for the data set I've shown you, so we have to deal with a heritability of 0.7. So that seems really feasible to exploit these genome-wide prediction approaches quite efficiently. So we tested the potential to predict grain yield using this large population of 4,000 input lines. And we performed a so-called 5-fold cross-validation scenario to estimate the prediction accuracy in this population for the trait grain yield. And it's very substantial. So we observe a prediction accuracy of 0.8, so which corresponds to around 60% of the variation, which we could explain by using these genome-wide prediction models. So which shows that this is very a good way to exploit genomics in breeding. We were also a little bit curious on whether we really exploit the knowledge of the genetic architecture much more efficiently in these genome-wide prediction approaches. And therefore, we took the same approach like previously in the genome-wide association mapping, and we split the population. So we took the 2,000 lines tested in the year 2012 and 13. We estimated the marker effects, and we independently estimated also the marker effects in the year 2014. And we correlated both effects, and the results are a little bit disappointing. So we end up with a correlation of 0.21, which is a clear indicator that we understand only a little bit of the genetic architecture of grain yield. But nevertheless, we can predict grain yield in a very efficient manner. And so the question arises, what are the forces leading to this high prediction accuracy? And one nice setup to investigate that is hybrid wheat breeding. We had a nice hybrid wheat data set, or we have 2 nice hybrid wheat data sets available, which allowed us to implement now a different way of cross-validation. So in these data sets, we had a set of male lines and a set of female lines, which have been crossed. And for this set of hybrids, we performed now a different cross-validation scenario. So we took a subset of male lines and female lines, and we looked at this number of hybrids, and we built up a calibration model, but we still left 10% of the hybrids in the data set to test our prediction models. And then by using this approach, we end up with 3 different test sets. So we have one test set where both parental lines have been tested in other hybrid combinations. So this is a T2 set. We have hybrids where only 1 parental components have been evaluated in the estimation set. And we have a completely unrelated test set where none of the parents of the hybrids have been part of the estimation set. So I use the same color code now to perform this kind of cross-validation. And you see now following results for 2 data sets. So we had around 1,600 hybrids available. and in the other data set we had around 1,800 hybrids available. And what you see, I have shown you also the results for plant type, but let's focus on the trait grain yield, is that as long as the estimation and test set are highly related, you do a fantastic job in predicting grain yield performance. So in that case, you end up at prediction accuracy ranging from 0.76 to 0.90 for grain yield. So you can do a fantastic job in prediction. But when you move to an unrelated space, you lose your prediction accuracy going down to 0.5 to 0.6 up to 0.2 to 0.3. So this is a clear indicator that the relatedness is one key driver of the prediction accuracies in these genome-wide prediction approaches. So now if you look in this data set in wheat, and if you think on the consequences for wheat breeding, you could also come up with scenarios where you exploit this relatedness. And you could think that if you have your estimation set and if you have different individuals you would like to predict, you could deduce out of the relatedness of that individual with your training set that if it's closer related, you do a better job in prediction compared to individuals which are less related, then the prediction accuracies should not be as high. And the animal breeders developed a very nice criteria to judge that kind of relatedness between the individuals which is going to be predicted and the training population. This is called the reliability criterion. And we used our data set to study the potential to exploit that kind of differences in the relatedness, to not consider prediction accuracy from the whole population, but rather consider the prediction accuracy at an individual plant level. So what we did we again used the 4,000 lines. We split it in two parts, and the 50% we used as a training set, and then the other 50% we used as a test set. And for the individuals of the test set, we used the genomic profiles to estimate more or less the relatedness of the individuals to the training sets, and we came up with different groups of individuals which are highly related to the training set or which are less related to the training set. And for this scenario, we again performed cross-validations and we studied the prediction accuracy. And this is the starting point. So if we look at the total population, we end up as a prediction accuracy of above 0.7. And now you see a clear trend. Yeah, you have subpopulations in your data set which are highly related to the estimation set, and for these subpopulations you do an extremely good job in prediction, so ending up in prediction accuracies of 0.96, which corresponds more or less to 50 location field trials. So this is a quality of data which is needed for variety release. So therefore, we propose that breeding companies are not implementing these genome-wide predictions for the population as a whole, but rather to exploit this reliability criteria and to adjust their breeding programs, that they work with different logistic strains for individuals where they know they can do a fantastic job in prediction. They can reduce maybe one step of phenotypic selection or intensive phenotypic selection, while other populations, they could not rely too much on these genome-wide predictions, and they have to be followed rather by intensive field trials later on. To summarize my talk, so I hope that I could convince you that the current population sizes we are working in plants enable only limited conclusions on the genetic architecture. So it's hard really to detect these small effect QDLs. I hope I could show you that the prediction is mainly driven by relatedness. And that has severe consequences, that it's not so easy to define kind of haplotypes based on the SNP profiles and then breed for these idiotypes. Third point is that if you implement these genome-wide prediction approaches, the prediction accuracies should not be considered for the population as a whole, but you need rather an individual basis of estimating the prediction accuracies. With this, I'm at the end of my talk, and I want to thank two persons from my group who contributed largely to the results. That's Dr. Yushen Zhao and also Dr. Zhang He, who performed most parts of the study. And I want to thank you for your attention, and I'm happy to take questions.
I'm pretty good. Question?
What is the system of pollination control that is being used in hybrid bees?
So all these hybrids were produced using different types of gametocytes.
What is it?
Different chemicals, gametocytes. So, and they were, I think, three different.
Are they very effective?
They are very effective. So we always control the sterility and the hybridity in the trials, and they are very effective.
Thank you. I think in many ways you just replicated what we've kind of over time found in maize. Obviously a hybrid crop, and we see very similar things. What I'm wondering is, have you come up with a way to sort of dynamically make these prediction sets on a, in our case, a population by population basis, or in your case, an individual by individual basis, to kind of automate that and make it simple to do? I mean, you sliced your by relatedness, which is certainly one way to approach it. But as you're looking at predicting multiple individuals across several populations, how can we do that? Really quickly.
This is a big challenge for the breeders. Yeah. So to do or to design breeding programs for every individual, that's not possible. But I think that you could at least think on 2 strains for prediction. So we thought also on how to implement it in breeding programs. And for the highly reliable part, you could really skip one phase of early yield tests, which you perform at around 3 to 4 yield trials, and you could directly go to this expensive yield test at 10 locations. So you could really decrease the cycle length by 1 year for this top fraction. Yeah. And somehow you have to define arbitrary the threshold. Yeah. But it's clear, so it's hard really to define this threshold and do some model studies to think of that. Yeah, and then the other fractions you replace also the first rough phenotyping with corresponding to 3 to 5 locations, and then you have to go again on yield tests, and it's not worth really to directly start with these broad yield trials.
Yeah, here.
Yeah, sorry.
The phenotypic data, ED data of these 4,000 lines which was generated, was it generated under a single environment, or it was taken over years from the breeding programs?
Mm-hmm. So actually, there's always a strong overlap of environments, and the big advantage is that this is all data from German wheat breeding, and we don't have a kind of strong genotype times location interactions. We have rather strong genotype times year times location interactions. So the year play an important role. So you cannot find cluster of environments, so that's rather random noise. So the phenotyping is, or the locations chosen is not so important for the results. Yeah.
It's really interesting to note that predictability is related with the relatedness that in your training population and other part. In that sense, if we do believe that we need to enhance the diversity of the breeding programs by pumping in the new diversity from some unrelated land races or other stuff in the gene bank, in that case, how can predictability enhance so that people start really not losing a kind of interest that, look, because I am putting it more germplasm, predictability will be low. Mm-hmm. So should there not be an effort to ensure that even if you put little limited diversity in the program, predictability is still at the acceptable level rather than nosediving?
Mm-hmm. So actually, this is an excellent question. So one point is, if you want to introduce really novel variation, it is not so good to rely on genome-wide predictions, but it's rather better to rely on field trials. Yeah, that's very easy. Yeah, you simply have to go this hard way. Yeah, but there's a second component. As we exploit mainly relatedness, breeders have to be very aware of not shrinking down their diversity too tremendously, because it is not relying on true knowledge of the genetic architecture. So you could lose valuable alleles if you focus too much on these prediction models. The big advantage is if you recalibrate every years and throw out old lines, then you have also quite a severe change in the marker estimates. So we have double-checked that, and that means that you have every year kind of different pressure on the markers, so it is maybe not so severe to lose diversity applying it.
Over here.
Will your prediction accuracy be improved by using multiple marker haplotypes rather than individual SNPs? You're getting a better indication of identity by descent rather than identity by state. Mm-hmm. And also, I presume, in a realistic breeding scenario, because the female side will be constrained, you'll have a smaller number of females. Mm-hmm. The problem won't be quite as bad as you make it out.
Mm-hmm. So it is easy to show, not easy, but you can show based on quantitative genetic theory that using haplotypes is nothing else than exploiting local epistasis. Yeah? And it is indeed the case in wheat, so that is also a published story, that if you shift from models exploiting only additive effects to models exploiting additive times additive effects, you increase the prediction accuracy up to 10%, which is quite substantial. And also that you increase it, but only slightly, if you move to the haplotype approach where you look at local epistasis.
I think you mentioned about the animal breeders, where they do A into B cross, and the F1 is actually a useful animal in the case of a dairy animals. And then they make a cross again. In the case of wheat, what you have showed, because there is not much genetic variation within the breeding program, you are not going to make significant advances. So one need to bring crosses right in the F1 population itself. That's where the genetic variation can come.
I didn't catch your question.
So the question is that you mentioned about the dairy animals, or the animal breeding population program. You cross A into B, and you get the F1, which is a productive animal. Then you bring another bull or artificial insemination and make the crosses, so you make huge genetic variation there. Whereas in the case of wheat, we wait till F5, F6 homozygous to make crosses. That's where we're losing the problem.
Okay, I fully agree with you. The animal breeders are closer to the breeding values, also with respect, so the average performance of their progenies compared to the population mean. And it's different in line breeding. It's very severe, so we always rely on non-additive and additive variation. In hybrid breeding, you're getting better, at least, I would think, yeah, that you evaluate a gamete and different genetic backgrounds, and I would assume that you are closer to the breeding value than looking at the line per se performance, but that is an intensive discussion going on, whether we could exploit all this variation and do maybe a better job in parental selection if we focus on the general combining ability effects compared to the per se performance. And you would indeed end up with completely different selection decisions, whether you go for per se selection or whether you go for GCA selection of the parents.
Great, that's a great discussion. Thank you.